And this is the first of our NANOvember lecture series. Thank you for coming. I'm going to talk about nanotechnology and aging, something that's very close to my heart as I age continually. But I'm going to start with this slide. Who knows who this is? You guys that are ringers can't say. Keith Richards, there you go. Now, he turned 65 in 2010. He's been looking like this since 35 years of age. Okay, so he's actually done something right. Okay, well, not really, but anyways, I point him out because this is what is happening to our population in the coming years. In 2010, the first baby boomers retired. Okay, and continually we get about 10,000 baby boomers retiring per day. So in the next 20 years, it's about 75 to 80 million people that are going to be above the age of 65. Okay? And this is just showing number of millions and percent of the population. And this is not something that's just occurring in the US. It's occurring worldwide. Okay? So what this is really doing is it's going to put a huge burden on our healthcare system. And to improve the quality of life of these individuals, to maintain their good health status. And so anything we can do to preserve, let's say, not necessarily make people live longer, but at least make people live healthier for their entire lifespan. And that's what the goal of some of our research is here at CNSE. So I'm going to introduce several of the theories of aging. The biological theories of aging. Okay, they're not all listed here. There's a number of them. I'm going to go over these in detail, but I think they all serve some function or another in the overall health of our society. Okay, the traditional wear and tear theory of aging. Okay, basically you start out, you're a beautiful '68 Camaro. I've always wanted this car, but I can't afford it. But after time goes by, you age, and you just wear and tear, the systems break down, and you no longer function, or can't function as vigorously. And so that's the wear and tear theory of aging. The autoimmunity theory of aging is one in which, as we age, our immune cells now attack our healthy cells. And so this is another component of the aging hypothesis. And actually there's some relevance in terms of this occurring. Okay? Well, our immune system now recognizes things that are normally considered part of our body as foreign. And we don't want that to occur. There is the programmed theory of aging. Okay, so this theory of aging encompasses the idea that we only have a finite lifetime. Okay, so we're all engineered differently. So for example, someone here on this timeline is going to give out in January of 2000, and that's it. And that's not really that credible because the average lifespan in this day and age is approaching 80, okay, in the US. The somatic mutation theory of aging is one in which our DNA gets damaged. And somatic cells are the ones that are not, we don't genetically inherit these mutations. They're acquired during our lifespan, for example, in response to ionizing radiation, in response to a carcinogen. And so this is another theory of aging. As we age, we accumulate this DNA damage. Then there's the homeostatic theory of aging. Okay, this theory involves the idea that we're no longer able to, our body's no longer able to supply the appropriate nutrients that we need to function. Okay, so this is what we refer to as the homeostatic theory of aging. And then one theory that's emerged in the last 10 years is the antagonistic pleiotropy theory of aging. This is a very sophisticated scientific term. What it actually means is things that are good to us when we're young, that help us survive when we're young, actually are bad for us as we age. And I'll give you some specific examples of that, and this is the kind of thing that we're targeting. There actually is some very good biological models suggesting that this is one of the key key theories of aging is actually holding true. And one that I like to study, and my favorite, is the free radical theory of aging. And my specialty is I'm a free radical biochemist, biologist. I look at free radicals. Does anyone know what a free radical is? I'll explain, but that's why we take antioxidants, to protect against these free radical species. Every food substance that you eat contains these antioxidants that protect against this free radical damage. Now, where this theory emerged was by this individual, Denon Harman, and it was back in the 1950s. He published this paper, a theory based on free radical and radiation chemistry. He's actually 96 years old now, so he's kind of figured it out. He's doing pretty well. He's been functioning. I read about him recently, and he was running 2 miles a day until he was about 89 years old. So I think that's pretty good. But the theory that he came up with was he started out as a chemist, and he was working on ionizing radiation. And ionizing radiation can cause a lot of damage. You guys need lights? Can you get those back there, Melissa? I think it's the last. There, is that better? So ionizing radiation produces a lot of free radicals. That high energy can modify chemicals and make them highly reactive. And a lot of this work was unveiled during the Manhattan Project during the 1940s, during the early war and during the World War II. And what they found out is that this ionizing radiation produces this hydroxyl radical that can damage biological tissue, okay, specifically. And what his theory was that, okay, so if there's this radical damage that's occurring, what if you were able to prevent these radicals with these reducing compounds? And these reducing compounds are antioxidants, and I'll talk to them in more detail. In this way, it might be a way to actually chemically prolong life and reduce the incidence of cancer in response to chemotherapy and in a nutrient-dependent way. Okay? So that's the free radical theory of aging. What's a free radical? So this is the only chemistry I'm going to give you guys, so don't worry. It's very simple. This is the oxygen atom. It has 8 electrons in its outer valence shell. What happens, a free radical becomes a free radical because it loses electrons in its outer valence shell, and it wants to when it loses an electron, it becomes very reactive. So it wants to pair with another electron. And so there's a wide variety of free radicals, and what ends up happening, it'll pull an electron from an associated chemical compound, and that chemical, no matter what it is, will become a radical in and of itself, causing a chain reaction with any chemicals that are nearby. So you get a lot of radicals, and this continues on and on. And the simplest example of this radical reaction is this here. Okay? When you bite into an apple, within minutes of you biting into that apple, you start to see this browning. And that browning is just radical reactions. It's the oxygen that's reacting with the lipids in the apple itself, causing this oxidation. So that's the natural process of these free radical reactions. And keep in mind, the apple itself is very good at protecting the inside of the apple from oxidative damage. Okay, so the peel of the apple is very rich in these antioxidants to prevent oxidation. So now we are continuously exposed to radicals throughout our life. Here's just some example. Here's the ionizing radiation can generate these radical species. In the center here, I have a picture of some nucleic acids, some proteins, some lipids. But radicals are produced as a consequence of smoke exposure. Nitric oxide is another radical species. Industrial pollutants in combination with ionizing radiation also produce a lot of radical species. Our own body can generate radical species. The energy-producing organelle in the cell, in the body, the mitochondria that makes ATP, generates quite a lot of free radicals. It's estimated that about 1 to 2% of all the oxygen that we consume on a daily basis is converted to these radical species in the mitochondria. the immune system itself, during the process of inflammation, has a system that generates these free radical species, and the reason for that is to kill invasive bacteria. So that immune system will generate these radicals to target viruses, to target pathogens that are maybe invading the body. So lo and behold, you wonder why we don't just sit here and spontaneously combust. Okay? We're continuously exposed to all these things. But we have a wide variety of antioxidants. We have all these defenders that come in and detoxify all these species. And I'm not going to go into all the enzyme systems that we have in the body. They're small antioxidants, but they all have some particular role in removing these toxic radical species. So all in all, this is what happens on the normal everyday basis. There's a fine balance between the antioxidants in your body and the radicals that are generated. Okay? And so everything's kept in check. It's when these things, there's an imbalance in the radical production and the antioxidant status that you now start to see disease processes occur. You see damage to proteins, you see damage to cell components, you see damage to DNA. Okay? In the last about 15 years, radicals have not just become damaging agents, they've actually played fundamental physiological, important roles in physiological processes in the body. And this is just an example of a signal transduction cascade. And what I'm going to talk about here is very brief. This is epidermal growth factor signaling cascade. And what happens in the cell, this is actually a process that's involved in a lot of cancers. This epidermal growth factor binds a receptor and turns on a signal in the cell. And that's all this is referring to. There's a variety of different cascades. And in the cell, there's about probably 1,000 of these different signaling cascades. And what this one promotes is cell division. And what does a tumor do? It divides uncontrollably. Okay, so that's just one signaling cascade. And what I like to make this as, I like to make this analogy is that this is somewhat like a circuit board. It's a cellular circuit where you plug in a signal, push a button, and you get a response. Okay, it's very similar. And it turns out that these radical species are involved in fine-tuning the regulation of this signaling cascade. So when there's too many radicals, a lot of these signals get turned on uncontrollably. And so one of the things that people are trying to work on is to deliver antioxidants to the sites of these signaling cascades to prevent some of these signals from occurring, for example, to prevent tumor proliferation or tumor growth. So stop me anytime if you guys have any questions. I'll stop right here because I'm going to switch gears a little. So those signals that I just discussed are involved in regulating a variety of pathways. One of those pathways is the process of chronic inflammation. And chronic inflammation has turned out to be at the crux of almost every degenerative disease process. So if you just pick one, skin disorders. As we age, our skin is not as functional, is not as protective. It actually doesn't heal as quickly. It's thought that inflammatory processes are involved in the function of the skin. In cancer, inflammatory processes that are partly mediated by free radical signals drive lung cancer progression, pancreatic cancer, colon and breast cancer progression, a variety of inflammatory disease processes. So, for example, chronic obstructive pulmonary disease, which is associated with chronic emphysema, arthritic disorders, inflammatory bowel disease, metabolic diseases such as diabetes and fatty liver disease. A variety of neurologic disorders are all associated with this chronic inflammation that is in part mediated by these radicals. Alzheimer's disease, Parkinson's, many diabetic complications such as sepsis, renal failure, neuropathies are associated with chronic inflammation. Lastly, cardiovascular disease. A lot of inflammatory processes drive specific diseases. They drive atherosclerosis, heart failure, and stroke injury itself. Okay, so if we can figure out ways to limit the production of these radicals at the site, we might be able to limit some of these degenerative disease processes, and that's the kind of thing we're working on here. So fortunately, we can actually study the aging process in a cellular system. We can take cells and culture them in our laboratory, and a lot of the things they do as they in cell culture, they mimic the aging process in vivo. So this is just an example of cells that are relatively young in age, and this is sort of chronological aging. And here's an example of cells that become senescent, or they age in a cell culture system. And we can model this in vivo, and it turns out that these senescent cells are produced in our body. And why are they produced in our body? Okay, what is the physiological role of cellular senescence in vivo? Well, it turns out that senescence is basically a cell that becomes damaged. It's programmed to all of a sudden become metabolically inactive, to sort of clear it from the system. So for example, when you're young, this is an example of this antagonistic pleiotropy. When you're young, a cell gets damaged. Okay, it puts out these signals that say, come and clear me out. So these senescent processes become very important when you're young, or they turn off that senescent cell that's damaged, and this is what happens when you're young. But as it turns out that a lot of these cells also stick around for a long time. And when they stick around too long, they're not necessarily good citizens anymore. They're not clearing the damage. They turn out to promote the damage. So this is referred to, and this is just a complex term, it's the senescence-associated secretory phenotype. So for example, cells in our body, they get old and they secrete proteins that aren't normally secreted. Some of these factors include a family of proteases that degrade their surrounding environment. Okay? So the cells become old, they start degrading the surrounding environment, they make other factors, growth factors, cytokines, interleukin-8, and a variety of interleukins, and there's master regulators of this process, and this is one of the things that we're studying in the laboratory. Turns out that these are all inflammatory markers, these inflammation-associated markers, and they're produced in abundance by these senescent cells in your body. So one of the ideas is if you can limit the ability of these senescent cells to produce these factors, you can actually limit disease. So this is actually some work that a graduate student in the laboratory that's been working on for the last few years, Donnie McCarthy, and I'm going to go over briefly some of the work he's doing. This is what happens when a senescent cell is close to a normal cell. So this is the process of tumorigenesis, where a tumor goes from its epithelial environment and transitions into a more metastatic, malignant, aggressive tumor cell type. And what happens is the things that are secreted from these senescent cells cause a normal cell to diverge into a tumorigenic cell and become malignant. So if you can limit the formation of these particles or these molecules that are coming out of these old cells, you might be able to limit disease or degenerative disease. So we've been working on, I'm not going to go through all the data, but we've been working on this, studying how this senescent phenotype works, and it turns out that it's driven by these radicals. Okay? The master regulator of the senescence phenotype is responsive to increases in radical production. This is one example of a radical. This is hydrogen peroxide. Hydrogen peroxide, you use it as an antiseptic at home. Okay? You see it oxidizes. When you put it on a wound, you see all that fizzing. That's because it's being removed by an enzyme called catalase generating oxygen. So it's very reactive. One unique feature of hydrogen peroxide, it's electron neutral. So it can actually traverse in and out of the cell, and it can go to various places in the cell. And it's used as a signaling molecule. It drives this master regulator and drives this phenotype. So what we're trying to do is target this phenotype and see if we can inhibit this specifically in these senescent cells. So last November, this paper came out from this laboratory, Jan van Derzen's lab down in NYU. And what he did, he developed a mouse model where he could knock out senescent cells. So he developed a mouse model that had this disorder. So it actually mimics the premature aging syndrome progeria. Okay, so these kids, they live to about 20 to 25 years of age. They prematurely age very rapidly. And there's murine or mouse models of this disorder. And what he did was to develop a mouse model where he could selectively kill all the old cells in that mouse. It's a really elegant genetically engineered model. But what he saw, this is the phenotype. This is the normal mouse, the diseased mouse, and you can see it has this weird syndrome they call leucopyrosis. It's when this back curvature. But in the animals where they knock out all the senescent cells, the mouse shows none of these phenotypes. Another thing that they saw, these mice didn't develop cataracts, and they didn't display any muscle atrophy or sarcopenia. Okay, so this was actually the first demonstration that these senescent cells are really important to the process of these age-specific disease processes. So this is some work from a medical student that was working for AMC. I think he's a third-year student now. But what we were interested in is finding out natural compounds that may inhibit this process. And so what this graph is showing is we're looking at one specific protein that's involved in this senescent process. And what you can see, these are young cells. They don't make much of this protein. But when they get really old, they make a lot of this protein. And what we were able to do is to screen some antioxidants and find some, and this was something that came from an apple peel extract, that completely prevented the ability of these old cells to make this senescent protein. Okay, so the goal now is to see if we can target this antioxidant for therapy. And the question you all want to ask is, why don't we just take general antioxidants for every day? Well, we do, but there's been studies that have met with miserable failure, I should say. I guess that's the only kind of failure you have, is miserable. But these antioxidant studies, actually, these were clinical studies done in Sweden with about 30,000 individuals. And what they saw was, actually in Switzerland, sorry, it's not Sweden, it's Switzerland. But anyways, what they showed is they were looking at lung cancer incidence in chronic smokers. And they treated these individuals with either vitamin E or vitamin A. And as you can see here, when they looked over the lifespan, the individuals that took the placebo did as well as the individuals that took the antioxidant. And in this case, it turns out that the individuals that took the antioxidant actually did worse. Okay? They had more cancers than the placebo group. They actually fed these guys enough vitamin A or beta-carotene, so the stuff that's in carrots, to turn these people orange. And I was able to get a picture of the test subjects. Here, this is the treatment group here. As you can see, they have this aging phenotype, the gray hair on their eyebrows. And this is the control group. Well, so the take-home message from this slide is that you can't just take a lot of antioxidants and expect to cure everything. You really need to target these antioxidants to where they're needed, and that's what we're working on. So one of the ideas that we have, and we've had this going for a few years, and this is one of the reasons I came over here, was to develop targeted therapies. So really encapsulate these antioxidants in particles that are only going to be released at sites of the disease processes. Okay, so we have done this. We've actually done this in the laboratory. And what we have done is develop these nanoparticles that contain antioxidants. And so these things are not going to be released all the time. They're only going to be released when they're needed. And so when they reach a site of disease, such as the site where these senescent cells are generating these proteases that degrade the environment, they're going to be degraded, and they're going to release antioxidants. And what those antioxidants will then do is then specifically target the free radicals that are being generated in these senescent cells and block that process. Okay, we've actually successfully done this. I'm not going to show any of the data, but this was things that we've done about a month back. And about a month back, a paper came out where they actually showed nanoparticles specifically targeted against aging. It's very similar to what we're doing, but this was published in this journal here, and this was in Science Daily. Basically what they did was they took particles that are only degraded at the sites of senescent cells. So the next step is to see if this can be used therapeutically. The reason for this is we can't genetically engineer human beings to get rid of senescent cells. We can only target them with therapies, and this is the kind of thing we're working on. And what they've done here is they did a little something different. They're able to take advantage of an enzyme system that gets degraded near senescent cells and then releases its cargo, so releases its therapeutic at that site. So this is a recent study using buckyballs. So I think the individual that invented buckyballs won the Nobel Prize about 10, 15 years ago. I don't know if that was that long. But anyways, there were some early studies suggesting that these carbon chain structures are very toxic. And so what this group did was to test the toxicity of these buckyballs in vivo. And they took the buckyballs and administered them with olive oil. This is a French group, so they're quite interested in the olive oil. So what they did was they administered the olive oil, and they looked at lifespan over time. So this is a survival chart, and this is the normal lifespan. If you look here, this is this number of survivors here. This is the age in months, and a normal rat lives to about maybe 24 to 36 months. Of age, approximately 2 years. When they treated with the buckyballs and the olive oil, they almost doubled the lifespan of these animals. Okay, and they were expecting to see these animals die, and all of a sudden nothing happened to these animals. This is olive oil alone, so you could actually just go home right now and put a lot of olive oil in your salad, and it might help you out. But the reason for this protection is that they showed that these buckyballs, for some reason or another, decrease the oxidative damage or the free radical production in these animals and extended their lifespan. Okay. Yeah.
What are buckyballs?
So buckyballs are these C60 fullerenes. They're carbon structures. These are just carbon molecules right here, linked together, and they form these chains. We work with a lot carbon nanotubes. I'm not sure if you've heard of. They're just long chains of carbon linked together, pure carbon. Graphite, lead in your pencils, pure carbon. It's just how the structure is formed. And the reason these came out and he won the Nobel Prize for this is because they also turned out to be very hard to make these, but they also can be important for carrier product, to carry things, because now you have a carbon structure where you could cage a particle in there. So where these things were worked on at first is they took these these buckyballs, and they can modify the outside of them to put certain proteins or target them for therapeutics, or load the buckyballs with a therapy themselves. And so that's where, but they just took these buckyballs alone. Yeah.
Wouldn't its mobility be decreased by its size?
Well, that's a whole question about why these things worked. Because you would exactly, I mean, these are fairly small. You got to think about these. These are probably in the 10 to 20 nanometer range. And I didn't give the traditional how small is small in the Nano College slide that we normally do. But there should be some limitation. I don't know. One of the thoughts here is that they probably cause other cells to engulf them, and they get. I personally think that they cause cells to get frustrated. Okay, so they start working on the buckyballs, breaking them down, maybe making more antioxidants. But you're exactly right. This is a really early study, so they don't really know how they protected it yet. They just looked at some of these antioxidant markers and showed that there's a big increase in a lot of the antioxidant enzymes. So one of the other things we're working on here is developing sensors for stress markers. So this is a project I'm working on with the defense contractor FLIR. They make thermal imaging devices, And what we're doing is engineering nanoparticles that have a variety of different constituents in them that will detect different analytes. So for example, this is an example of one of the nanoparticles that we're working on that will detect glucose, urea, creatinine. This is something that's released from the liver when it's damaged. C-reactive proteins. All of these are stress markers that are released in your body, and we're making nanoparticles. And the reason we're doing this, a defense contractor is interested in this, is they want to be able to know when their war fighters are stressed. Okay? And if we can develop these particles to sense the stress in these warfighters, we can maybe prevent when they're exposed to some toxic agent. If they're exposed to a gas or something like that, we may be able to detect that very rapidly and treat them instantly. And the reason this imaging technology company is very interested in is they make a device that can sense these particles. So the idea is to wear some type of a wristwatch that can sense these stress markers. It'll automatically be read and tell you when there's some type of stress or disease or inflammation going on in the body. And this is one of the things we're working on. We just currently got funded to develop these particles. So the reason I mention that is a lot of what we learn, what we benefit from in society, it comes from the Defense Department. I think we all know about where the internet came from. And so this is another example of hopefully what will emerge from this. This is something we're working on in the Nanobioscience Constellation, sort of making a chip circuit that can detect biomolecules. And that's one of the big hurdles in the industry right now, is that how can we develop a sensor that can detect markers in the body on an electrical chip. Okay, remember electricity does not function well in a liquid environment. Okay, so we need to be able to develop a sensor that has a biologic device in it to analyze and sense the environment, linked to an electronic chip that will develop a signal. And so in this process here, we have an interface for sensing and drug delivery. We could actually, this device may sense inflammatory marker. It may also deliver a drug at the same time. We have to develop a compatible layer that will not foul the whole system. And this is the things that we're working on in the college. Now, can this actually be done? This actually came out of a recent paper in Science in September of this year. And they made actually a dissolvable electronic chip. So this is an electronic chip made out of silt with circuitry in it that can be put into the body and eventually will decompose. So who wants to put a chip in their body? I don't think anyone here would raise their hand and say, line me up. But these things will actually degrade into non-toxic substances. So I think the future is close where we're going to be able to not only develop particles that can detect damage in our body, we can target damage in our body, but we can also develop sensors and electronics that can therapeutically treat that damage. So I like to put this slide together. I sort of adapted it from this website here, but you can think about what's going to be happening in the future, and I think this future is fairly close, is that we're going to be developing electronic devices that sense our health status. That health status can be delivered to your own home gateway, for example, your home computer, a mobile device, your smartphone, and this can be under your control. You don't want all your health statistics going out willy-nilly into the internet. Okay, you want to be able to control some of this stuff. So this is where this is controlled. If you want to say, okay, my PDA is telling me that I'm not feeling that well and I should go get this drug therapy, then now you'll let the people that you need to know. Your home health care specialists, even your friends. And if it becomes a bigger problem, you might want to feed that information to your physician. And this can all be kept and tracked on a sort of an electronic health record. Now, one of the important things about this electronic circuitry and this sort of nano revolution is that you can think of other things that this can do. This can also speed how quickly we get drugs to the clinic. Okay, the problem now with clinical screening of drugs is you need to treat patients. You got to go through phase 1, phase 2, phase 3, and it takes forever to get these drugs through the clinic because you need to look at responses, long-term responses. If we have biologic sensors that can tell you when a drug is failing early, then we can pull that off the market, and the drug companies don't have to spend billions and billions of dollars screening a variety of different drugs. Another thing you can think about with these types of sensors is that, for example, if every one of us in the room has one of these sensors implanted or a biologic device, we'll know when there's, let's say, a flu pandemic. So the local authorities will be warned, okay, there's a population of individuals in the NanoFab South auditorium that are kind of feverish because of, including myself, that need to be treated or could be a problem occurring. And so this can keep a close eye on pandemics, emerging pandemics in society. You know, new flus, new bacteria that are emerging, and things of that sort. So I have to thank all the people in the lab that work for me, that do a lot of this work. Past members of the laboratory, current members of the laboratory, a lot of my collaborators, and my funding sources.